So many of the initial symptoms that you've heard these families talk about are the same symptoms that Duchenne described in 1861 when he saw his first patient, Joseph Sarrazin. So Joseph was born without any difficulty moving his limbs, and he appeared to be completely normal. However, he walked later than his brothers, and once he began walking, he had an abnormal gait where he would walk with his legs spread far apart. At 3 years old, it was noticed that his lower extremities grew in volume. When Joseph came and saw Duchenne, Duchenne performed a physical exam and noticed that the muscles in his lower extremities were larger, and they were quite firm to touch. He noticed that Joseph didn't like moving his lower extremities, and when he did, his movements were weak. He noticed that his walking was laborious. However, he also noticed that his upper extremities appeared to be completely or relatively normal. He also noticed that Joseph had a dull intellect and difficulty speaking. After seeing several patients with these same clinical criteria, Duchenne developed some diagnostic criteria for this condition, and he had 6 features. The first feature was that there was decreased strength in the lower extremities, that these patients had lordosis, which is a curvature of the lower spine, and that they had difficulty when they walked. He noticed that they had excessive development of volume or hypertrophy of the lower extremities, and that this disease worsened until there was complete general weakness. He also noticed that this was coupled with decreased electromuscular contractility. And importantly, he noticed that this was in the absence of other things that would suggest peripheral or nervous system abnormalities or infections. So Duchenne called this disease pseudo-hypertrophic muscular paralysis. And one of the interesting features of his name is he says pseudo-hypertrophic as opposed to hypertrophic. And this is because when he looked at a biopsy specimen of these patients, he noticed that instead of having classical hypertrophy of their muscles, they had something else. But before we get into what the histopathology looks like, I just want to teach you quickly about what the normal muscle histology looks like. So muscles, if sectioned in cross-section, have a nicely arrayed units that are single muscle fibers themselves, which are single cells. And an individual muscle fiber cell is a multinucleated cell. One of the interesting features about being multinucleated is that if a segment of the muscle were to have some degeneration or death, the neighboring segments in that cell can remain alive with the nuclei that they have within them. Muscle fibers also have an interesting cell membrane structure. The outside of the cell membrane is called the sarcolemma, and there's invaginations of the sarcolemma that form these T-tubular structures. And this membrane branches extensively over the contractile units in the muscle fiber, which are called the myofibril. And if you were to look by electron microscopy at a myofibril, you'll notice that it's composed of multiple repeated arrays of something called a sarcomere. And a sarcomere, the 2 main proteins in a sarcomere are myosin and actin. So the thick filaments in the sarcomere are myosin, and the thin filaments are actin. And when you look by electron microscopy, you can notice banding patterns of these proteins. So the end of the sarcomeres are called the Z disc. And you can see here that myosin is connected to a band called the M disc in the middle of the sarcomere, and has connections to the Z disc at the end through a protein called titin. And actin itself has interactions with the Z disc, and myosin and actin can interact, and the myosin heads will pull on the actin when contraction occurs and move the Z discs together. So this contraction that I just described to you is a calcium-dependent process. And so the way that this would work is that if you had electrical activity coming from a nerve that would depolarize the muscle cell membrane, that depolarization would propagate throughout the T-tubule system, activating voltage-dependent calcium channels within the membrane there. These channels will also activate channels within a membrane structure in the muscle called the sarcoplasmic reticulum in order to release calcium from the sarcoplasmic reticulum and allow calcium to flow in through the membrane. The rise of intracellular calcium will also initiate the contraction of the sarcomeres within the muscle. In order to stop contraction, the intracellular levels of calcium have to be removed. And in order to do that, you can put calcium back into the mitochondria, back into the sarcoplasmic reticulum, or out across the membrane. And the decrease in intracellular levels of calcium will then initiate the relaxation of the muscle. So there are 4 major muscle fiber types within skeletal muscle. There's type 1, which is also called red fibers, type 2A, which are also called red, type 2X, and 2B. And these fibers are mainly determined by the types of myosin that they express. So you can see here that all these different fiber types are expressing different types of myosin. And the type of myosin that expresses will determine the kinetics of how that muscle contracts. So type 1 fibers have a particular form of myosin that has a slow contraction time and produces low levels of force. You can stain for these different types of myosins in the muscle, and if you look at the cross sections that I showed you of muscle fibers, you can see that they're arranged in a mosaic pattern within the muscle. Additionally, there's other interesting features that distinguish these muscle fiber types. So, and that's mainly based off of their metabolism. So type 1 fibers, or red fibers, have, like I said, are slow to contract, and they're mainly used for aerobic activities. They have a very high mitochondrial density, and they mainly use oxidative metabolism. In stark contrast to them, type 2B fibers, which have a strong contraction, are mainly used for anaerobic activity. They have low numbers of mitochondria and mainly use glycolysis for energy generation. So now getting back to the muscle histology that Duchenne would have seen in his patients, there are 3 characteristic features of the muscle histology in Duchenne's patients. You get large patches of muscle fibers that are necrotic, and these patches are invaded by phagocytic cells. So that's why the appearance here looks to be very dark. Additionally, you can see in this image down here, you have muscle fibers that have the nucleus within the center of the fiber, and normally they're in the periphery. And this is indicative of regenerating muscle fibers. And lastly, you have interstitial muscle fibrosis. And additionally, if you were to look at the ultrastructure of the sarcomere itself in a Duchenne's patients, you can see that the normal banding patterns and structure of the sarcomere is abnormal. So the key facts that we want you to remember from this initial description of the disease is that Duchenne's muscular dystrophy is not a developmental disease based off of the clinical presentation, but instead is thought to be degenerative. And if you look at microscopic specimens of patients, you can see that there are many necrotic fibers and other fibers with abnormal sarcomere structure. So a key question to answer would be, is this disease genetically inherited? And if it is, what is the gene that's responsible for it? And it was known that Duchenne muscular dystrophy primarily affected boys, and it in fact affects about 1 in 3,500 boys. And this type of pattern suggested that the gene was linked to the X chromosome. And so in studies in the mid-90s, they had identified that using cytogenetic chromosomal banding patterns, you could identify large deletions in Duchenne's patients that mapped to a region of the X chromosome called P21. And in 1985, Louis Kunkel used a subtraction cloning method in order to positionally clone the gene, and he identified a cDNA fragment that was missing in Duchenne's patients. And once they were able to identify a cDNA fragment that was missing, they could generate an antibody. And once an antibody was made, it was off to the races. And very quickly, after generating this cDNA fragment, they were able to identify that dystrophin was associated with other proteins that they called the dystrophin-associated protein complex. They also identified that dystrophin localized to the sarcolemma of the muscle cell. They found out that it interacted with F-actin, which is a cortical cytoskeletal actin. And they also found out that other members of the dystrophin-associated protein complex interacted with extracellular proteins such as laminin. By 1993, they had a model of what dystrophin was doing in the cell, and they had shown, and the model was that the N-terminal portions of dystrophin interacted with cortical cytoskeletal proteins such as actin, and that through its interactions in its C-terminal domain, it interacted with a transmembrane protein complex that had extracellular glycosylated proteins that interacted with extracellular matrix proteins. So since 1993, there's been a lot of work that's been done on this protein complex, and there's been many other proteins that have been added to this picture. And interestingly, many of these proteins are also associated with different types of muscular dystrophies. So for example, other members of this complex called sarcoglycans are all associated with different types of muscular dystrophies called limb-girdle muscular dystrophies. And I think that this really highlights the key importance for this protein complex in a muscle cell. And so just to add one more of the key facts that we want you to remember from this introduction is that besides muscular dystrophy is a degenerative disease with abnormal muscle structures, and importantly, there are many muscular dystrophies that are linked to a protein complex in the muscle cell membrane. And so really what the important question that the field has tried to address is what the function of this complex is in the normal muscle, and how does its deficiency lead to degeneration.
Okay, this is working. So I think this raises the big question of how do we figure out what the function of a protein is, and especially what the function of the protein is in the context of a disease. And so as Kevin said, the earliest studies that looked at dystrophin, and dystrophin is indicated here in purple, looked at dystrophin in the context of analogy to other proteins, and they saw that, okay, there's an N-terminal domain that looks like other N-terminal domains that bind actin, so it probably binds actin. There are some spectrin repeats that look like a protein called spectrin. That's an important protein in comprising the cytoskeleton of a cell, giving a cell its shape. So this looks a lot like spectrin, and it purifies with other proteins that are associated with the membrane. So for the most part, this looks like a membrane protein, and it probably has some important role in the membrane of the cell. And so when they look back to older studies looking at histology of Duchenne's patients, this kind of made a lot of sense. So if you just look at kind of a microscopic image of a cell from a Duchenne patient, these guys, Andrew Engel's group, saw that they have these delta lesions, and it looks like a subplasmalemma lesion where the plasmalemma is fractured, and you could see this on EM. Here's the plasmalemma here, and there's this discontinuity here. And so what they concluded was the primary defect in the disease was a lesion of the plasmalemma. And so taking it further about 10 years, H. Lee Sweeney's group said, okay, well, if the primary defect is a sarcolemmal defect, and we could assess this by looking at permeability of the cell to a membrane-impermeable dye. And so they did these experiments where they took diaphragm from either a wild-type normal mouse or an X-linked muscular dystrophy mouse, and they tried to stain them with a membrane-impermeable dye. So normally this dye shouldn't enter the cell. And so if you look at normal mouse, you see no permeability to the dye. However, in the MDX mouse, you see a limited amount of permeability. Now, if you hypercontract the muscle and thereby stimulate some damage to the muscle, in the normal case, you get some permeation of the dye into the cells. And in the muscular dystrophy mouse, you get a lot more. So this suggested that the primary defect here is one of membrane fragility. And this is kind of the most dominant idea right now, is that loss of dystrophin produces membrane fragility, which results in membrane damage, and somehow this leads to myofiber necrosis. And that somehow came back to earlier work that showed that essentially what happens in the cell is if you have damage to the membrane, then calcium can influx into the cell. And so what I'm showing here is again from Andrew Engel's work, staining the tissue with a dye with two different reagents that stain calcium. And so if you look at normal tissue, you don't see the stain. But if you look at tissue from Duchenne's patients, then you see that the cells readily stain for calcium. So this suggested that there was intracellular calcium uptake in these cells. And their conclusion from this paper was that it was via the membrane lesions. And so this gave rise to the model that the current understanding of Duchenne muscular dystrophy, and that loss of dystrophin produces a membrane fragility phenotype. Membrane damage allows calcium influx. This activates proteases that are activated by calcium that degrade the cell, and this leads to myofiber necrosis. Okay. So now there are other, so that's one model. It's the most dominant model. That's what you'll read in the literature. But there are other models that are starting to emerge, and especially in terms of calcium. So other groups have begun to look at calcium in the cell using electrophysiology, and they found that there are various channels that may be associated with increased calcium in these cells. And so when they compare calcium fluxes in normal versus muscular dystrophy mouse muscles, then various groups have reported different channels that might be present to allow calcium in. And there really isn't any consensus on any one channel that is doing this yet. There's a lot of literature on different things, but essentially the way that model goes is a little bit of a flip, that you have aberrant channels that allows calcium into the cell that activates proteases, and then you get myofiber necrosis and membrane damage. Okay, and so the last model, which comprises many models, and I'm just going to kind of summarize this for lack of time, involves signaling cascades. And so a lot of the more recent work on Duchenne's, and especially in mouse models that have been missing many different components of the dystrophin-associated complex, have found that, in fact, many of these proteins associate with signaling proteins that we understand very well and that we know are involved in cell survival cascades. So it's possible that, in fact, loss of dystrophin, and therefore loss of the dystrophin-associated protein complex, results in loss of this scaffold and essentially decrease of these cell survival cascades. And so this is another model that's under active investigation at the moment. Okay. So as we started reading this, we were really unsatisfied by these models, primarily because, first of all, they didn't seem to explain any of the pathology of the disease. The primary model, the membrane fragility model, is essentially based on the assumption that myofibers take up a dye. So they're doing kind of a live cell stain in vivo. And we know that dead cells take up a dye. That's not new. So the question becomes, are the cells taking up the dye because they're dead, or are they taking up the cell because they have a fragile membrane? And furthermore, if you look at the course of the disease doesn't affect all muscles equally. There are many muscles so there's a progressive course of degeneration, but there are also some muscles that are spared, for example the extraocular muscles. And so the model that dystrophin is an important part of all muscle cell membranes, and that loss of dystrophin results in membrane fragility, doesn't explain kind of the more important nuances of the disease, I think. So as we started preparing the presentation, we asked the question, well, if we go back to the pathology, can we get any clues about what the molecular pathogenesis is? So now if you look to a review that's been published in the last 10 years, then you'll find that on histology, there are three very characteristic features of Duchenne muscular dystrophy. You see necrosis, you see cells with central nuclei indicating that the cells are regenerating, and you see inflammation. So we started with those and we just threw them out. What did I do? Yes, we threw them out. We said, okay, these are very non-specific. If you have a degenerative process, you're going to see these things. This is not interesting. Now, if you go back to some of the older papers and you look at some of the older pathology textbooks, and you read about some other things. So you read about hypercontracted fibers, segmental necrosis, and group necrosis. And I'll talk about each of these in the coming slides. Group necrosis is shown here, where a group of muscle fibers is closely associated with each other, is undergoing necrosis in the same state. So all these fibers are dying and they're being infiltrated with immune cells. Okay, so let's start with hypercontracted fibers. So these are samples from 2 patients with Duchenne. And I'd like you to focus on these very large cells. So these are large, opaque, kind of glass-like cells. They're called hyalinized. And so these are characteristic hypercontracted cells that you would see in histology from a Duchenne patient. And these are 2 different patients. And you don't see these in many other muscle fiber diseases. Now, if you look at these hypercontracted cells under EM, so this is a normal EM, and Kevin showed you what these look like. And so here's the Z disc, where essentially the actin filaments are attached, and then here's the center of where the myosin is. Now, if you look at these hypercontracted fibers, and it's hard for me to actually see this from there, what you see is the Z discs are essentially collapsed against each other. And the myosin filaments, they're so collapsed that the myosin thick filaments are actually curled up against the Z disc. And this is due to excessive hypercontraction of the muscle. And now you see this disorganization of the Z discs. The Z discs are usually very parallel under EM. And so you have a lot of damage to the muscle fiber here. How does this happen? So if you take a piece of muscle from a mouse with muscular dystrophy, an MDX mouse, you can elicit this by contracting it. And so here's a normal muscle on the left, and these are contraction episodes. So this is first contraction, and then subsequent contraction up to the 9th contraction. And this is the force generated. And you can see the normal muscle, if you stimulate it, it gives you the same amount of force. It gives you a stereotype amount of force with each contraction. However, in the muscular dystrophy mouse, the amount of force, first of all, the amount of force decreases with each subsequent contraction. And now if you look at the muscle itself, you see this blebbing starting to develop. So these are contractures that are caused by the hypercontraction of muscle fibers. And with each subsequent contraction, the force decreases because you're losing muscle fibers that are contributing to the contraction. So now if you zoom in on these hypercontracted regions, you see essentially what you see in the histology of Duchenne. So this is an example of 3 muscle fibers. This is a fiber that's been hypercontracted. This is the hypercontracted region, and this is another hypercontracted region, and if you slice it, you get the glass-like hypercontracted fiber that you see on H&E. Now if you do a slice through the center, you see a degenerating fiber. And so this is termed segmental necrosis, because you have a long muscle fiber and one segment of that is degenerating. So phagocytes will come in, they'll clear the debris, and then muscle stem cells will come in and repair this. If this segment happens to be the part of the segment where the neuron is innervating the muscle, then this muscle will need to be re-innervated by a new neuron. Interestingly, when this is reproduced in vitro, or not in vivo in rabbits, which is what they did in this paper, they reported that actually the only muscle fibers that they found hypercontracted were type 2B fibers. These are the glycolytic fibers. 8% were type 2AB, which are also glycolytic, but have some oxidative capacity. So these are fast twitch fibers. The fast white fibers are the only ones that are hypercontracting. Slow fibers are just fine. So is there any difference in fiber type distribution in Duchenne patients? The work from Helen Blau's lab at the end of the 80s showed that, yes, in fact, if we look at Duchenne patients, we do see differences in fiber type composition. And so here's a stain that's labeling all the fast fibers black. And you can see, so these are marked in B, so these large, dark labeled fibers, which are hypercontracted and degenerating fibers. And now if you use an antibody that will label all the isoforms of myosin except the ones in the 2B fibers, that is, so this is labeling everything except 2B fibers, then you never see labeling on degenerating fibers. So their conclusion was that, in fact, so in this patient who was 1 year old, every fiber that was degenerating was a 2B fiber. These are the fast twitch fibers. And in fact, if you look at later stage disease histology, then you do see this. And so here, the type 1 fibers are labeled dark, and the type 2 fibers are labeled light. And you see this predominance of type 1 oxidative fibers. And in fact, the very few type 2b fibers, glycolytic fibers that you see are like hypertrophic and probably degenerate. So interestingly, a lot of recent work has shown that, in fact, you can rescue the disease through any manipulation that promotes type 2 to type 1 fiber type switching. And I'll show you one example of this. For example, let's see, PGC. So PGC1 alpha, it stimulates mitochondrial biogenesis, and I'll show you that data in a moment, and that can rescue the disease. Calcineurin NFAT signaling, activated calcineurin NFAT signaling stimulates production of type 1 fibers, and if you activate calcineurin NFAT signaling in the context of an MDX mouse, then you rescue the phenotype. And here's some data from Bruce Spiegelman's group showing that, okay, if we overexpress PGC1 alpha in all the muscle, you get this very dramatic fiber type switching. So you've got the white muscle completely turning red, so all this muscle is turning completely oxidative. And if you do this in the background of the MDX mouse, you rescue the pathology. Okay. So I think the take-home point that we'd like to send right at this point is that we think people aren't spending enough time looking at the fiber type specificity of the disease. And there's a lot of good data showing that, in fact, type 2b fibers are much more susceptible to injury in the disease, and for some reason or another. And there's evidence showing that you can rescue some aspects of the disease if you promote the development of type 1 fibers from type 2 fibers. Okay, so then the next aspect of the disease histology that we'd like to address is group necrosis. And at this point, we're kind of speculating and putting this together, and there's not as much out there on this. So as I said, one of the things you see is the generation of groups of muscle fibers that are located very close to each other. And you wouldn't really expect this, because in general, the muscle is innervated by, the innervation of muscle fibers is distributed kind of in a checkerboard pattern. So these, for example, these muscle fibers wouldn't necessarily be the same type, and they wouldn't necessarily be innervated by the same neuron. But one of the places where you do see this is in a phenomenon called chronic denervation. So if you cut the nerve supply to a muscle, then you see group necrosis. So I'll explain to you how this occurs. As I said earlier, the muscle fiber type identity is determined by the motor neuron that innervates it. So if you take these two fibers innervated by different neurons with different types, and you deinnervate this muscle fiber, what's going to happen is this motor neuron is going to sprout a collateral, and it will innervate this muscle fiber, and now you'll get a type switch, where this muscle fiber will now become the same type as the other. And now, in a sense, you have a small group of two different types, or two of the same type. And when this occurs during chronic denervation, so for example, if you cut the nerve to a muscle, then I want you to focus on this green neuron here, and it's innervating all these white muscle fibers. So if you cut this, then you're going to deinnervate these muscle fibers, and they're going to get collaterals from this blue neuron that's innervating the yellow. You're going to get a type switch, and so now you have kind of this larger group of yellow fibers due to the reinnervation from this neuron. And so if you look at the muscle in chronic denervation, you lose this checkerboard pattern, and you produce these fiber type grouping patterns. So how could this happen in Duchenne? Well, as I've suggested, the pathology could be due to hypercontraction of muscle fibers. For hypercontraction to occur, you need a stimulus from a motor neuron. So if an initial stimulus results in hypercontraction of all of these 4 muscle fibers, producing segmental necrosis, loss of the end plate, and the muscle fiber becomes reinnervated by a neighboring motor neuron of the same type. You will now have a larger group of muscle fibers that are being innervated by the same motor neuron. And now if you get a subsequent stimulus that's going to produce a hypercontraction, now you'll get degeneration of a larger group. So with the progression of the disease, you could produce these larger groups of motor neuron muscle fibers. So based on everything I've discussed, this is the model that we've proposed and come up with for the pathophysiology of the disease. There's a question mark here that we can speculate on in a moment. The primary defect has to do with type 2B fibers and hypercontractility of type 2B fibers, which produces segmental necrosis and degeneration and deinnervation. Then you get potentially reinnervation of these fibers by a neighboring motor unit, forming a larger group, and this process repeats, and you can see this group necrosis. So we think this is very interesting because essentially most of the therapeutics at this point have been focusing on the gene, dystrophin. How can we repair the genetic defect? And everyone's essentially assumed that the function is just this general idea of membrane fragility, and the muscle cell membrane is fragile to contraction, and it's just being destroyed. We can't do anything there. But if we take this model a little bit further, what it suggests is that maybe there are positions where we can intervene pharmacologically to make the disease better. And so, for example, right here, there are many pharmacological approaches that could be used to potentially target fiber type switching. And if we think about the pathological process of the disease as being downstream of hypercontractibility, then we can begin to look at actual potential channel blocking agents or things that control muscle fiber twitch. And there are many of these drugs as well that could potentially be used. Okay, so this is my last slide, and this is kind of, the next slide says discussion, and this is just what we're hoping for in terms of discussion, are ideas that people have either that relate to the model we've proposed or perhaps relate to other models, or I don't know where this is supposed to go, but we'll see how it goes. So one potential idea relating to our model, and this is a recent paper that came out, was that, okay, so if you have hypercontraction, that suggests that the muscle is receiving a stimulus to contract, calcium is coming in, it's going to start contracting, but maybe it's not resting. So how does the muscle rest? How does calcium get removed from the cytoplasm to turn off the muscle contraction? Well, there are these pumps called Circa pumps. They're ATP-dependent calcium pumps, and they're present in the sarcoplasmic reticulum, and they pump calcium back into the sarcoplasmic reticulum. And so now if you overexpress these Circa pumps in the background of a muscular dystrophy mouse, then you can rescue the phenotype. And so if you actually look at the physiology of what's going on, essentially you're restoring, you're restoring the calcium flux back into the SR, and you're decreasing the muscle contraction. So there are drugs that could potentially do this as well. This is one area that we thought. Want to add to that?
Yeah, I guess the only other thing I wanted to add was when we were looking at a lot of the literature, we noticed that a lot of the studies have been done by like pooled muscle studies, or even if you were studying an individual fiber itself, people never take into account the particular fiber type that they're looking at. So let me give you an example of how this could be problematic. There's a lot of literature that said in Duchenne patients and in the MDX mouse that you get an increased resting level of calcium in a muscle fiber. And let's say this, and they say that this calcium level is increased about 2 to 3-fold. But if you just simply look at the difference in resting levels of calcium in type 1 versus type 2b fibers, you find that type 1 fibers have an increased resting level of calcium that's twofold. And now we've told you that if you look at the histology of a Duchenne's patient, they primarily, and the mouse, have type 1 fibers. So if you're doing an experiment where you isolate fibers from a Duchenne's mouse or Duchenne's patient, and you compare it to a control wild-type mouse or wild-type patient, you're actually comparing the difference potentially between just type 1 and type 2b fibers. And you know, you can go beyond this and say if people are just doing Western blots or looking at signaling pathways and just looking at the difference between an MDX mouse and a control mouse, you could be looking at differences that aren't really significant to how the pathology of the disease is occurring. And so I guess this is another point that we wanted to just emphasize, that I think that people really need to begin to pay attention to the actual cell types that they're considering when they're doing these studies. So yeah, I think